Okay, so let's go ahead and get started for today. So first, I know that there's one more class left after the Thanksgiving break, but I'll remind you that the final exam is Tuesday, December 10th. So it's like the Tuesday of not the week right after we get back. But I would like to suggest that you guys take the opportunity of the Thanksgiving break to, in addition to eating turkey and watching football, catch up on all the stuff that we've been doing these past couple of classes. So remember, because it looks like we're doing a lot of chapters here, but look at the stuff I've posted online about the readings. I explicitly tell you which sections to skip. Look at the slides we've talked about in class. Anything that we talk about in class is fair game, but I'm not going to ask you questions about stuff that's in the book. Especially on chapters 16 and 17, transcriptional regulation in prokaryotes and eukaryotes. There's way too much stuff to cover in this class, so I have carefully picked what we should go over. So you need to make sure that you know very well the stuff that we've talked about in class. However, the rest of it, not so much. So because I forgot to send you out a remind about the lab homework, which opened on it, I had it open automatically on its own, and it was supposedly due yesterday, but it's not. I sent you another announcement. I'm having that due Tuesday, the Tuesday we get back, but at the 12:30 a.m. So Monday, December 2nd, like overnight that night. And that is when the last the exam homework is due as well. So a couple of the questions, as I indicated when I posted it on Tuesday, for the lab homework are, we're getting to it today, but I thought it was important to include that material on there. All right, any questions before we get started? Okay, so here's where we left off last time. So we had talked about the four different kinds of operons, and I had said that the easiest way I know to remember it is that you break it down into two classes, inducible and repressible. Remember, we talked about this. And inducible is always off, but can be turned on. Repressible is always on, but can be turned off. And then for each of those, there's a positive and a negative. And we said positive means that the protein involved, or the transcription factor that binds the DNA, is going to be an activator that's going to turn it on, and negative means that a repressor is going to turn it off. And we ended by talking about the negative inducible operon. So that's where I'm going to start again, go over this, and then go over the other three classes. Remember that operons encode multiple genes that will usually give enzymes that are all involved in a particular process. And we talked about this is important because it allows bacteria an efficiency in that they only have to turn on transcription one spot, and then they get three proteins as made out of it for the I think the aliens are coming. For three proteins for the price of one. Also, whether it's a transcriptional repressor like shown here or a transcriptional activator, it's important for you to understand that the regulator protein is coming from a separate gene. So here's the operon promoter operator that we talked about last time, where the repressor is going to bind, but the regulator protein is going to come from a different gene somewhere else in the genome. Generally, because these are allosteric, shape-changing proteins, this transcription is going to be on all the time. So whether it's an activator or a repressor, this regulator protein is always going to be around. All right, so back into it. Here we have a negative inducible operon. So this is always going to be off. And at the end last time we said that's because under normal conditions, the regulator protein here, which is a repressor, will bind to an operator sequence. So it recognizes the DNA sequence, just like the restriction enzymes that we use to cut the DNA in the lab class. But instead of cutting the DNA, the repressor protein just sits there. And the way that it represses transcription is it's occupying a spot that the RNA polymerase, led in by sigma subunit, is trying to get to. And if the repressor is there, the RNA polymerase can't occupy the same spot and transcription doesn't happen. So that's the default state. That's why this is an inducible operon. Then if the inducer molecule comes in, it will bind to this second site up here on the repressor and it will cause an allosteric change, or a change in the shape of the protein. If the inducer binds to its spot, that makes it so that the repressor can't bind to the DNA and it pops off, allowing RNA polymerase to bind and transcription happens. So if the repressor didn't exist in this case, if you had a mutation in the if you had a mutation here that made a repressor that was unable to bind DNA no matter what, this gene would be on all the time. But it's off because the repressor is usually okay. All right. And we said that in the case of inducers, generally they're going to be a molecule that starts at the beginning of a biochemical pathway. So this could be an example of a biosynthetic pathway where the end product W over here on the right, is a nucleic acid, an amino acid, whatever you want to say that the bacteria are building. The precursor, by being present, indicates the need for the enzymes that do the three steps shown here that convert that precursor into the intermediates and then finally into the product. And so the more of the precursor that's there, the more of the repressor protein that is bound up and therefore unable to bind, and the more, lack of better words, the more on the operon is, right? And I mentioned last time that people tend to think of transcription as being like a light switch, where you're either on or you're off, but I like to think of it as like a dimmer switch. So you can turn it almost the whole way off, and in some cases all the way off, or any level of on or off. And I talked to you about chemistry again last time. We talked about stoichiometry, right? There's a certain amount of the repressor protein always there, and then the amount of activator or inducer there is going to determine the amount of repressor that's bound or not. So if there's a lot of the precursor protein, the repressor is going to be completely bound up and the gene's going to be on all the way. But as this precursor gets used up in the biosynthetic pathway, if no more precursor is present, then you want to downregulate these genes, because bacteria are always in a competition for their life, right? They're trying to replicate as fast as they can, and all the other bacteria in that same environment are doing the same thing. And so any waste making enzymes that you don't need is going to hurt you. Okay, so this is the negative inducible. Now let's look at the negative repressible. So because I said negative, it's still always going to involve a repressor protein there. But because it's repressible, this operon is always going to be on unless the status quo is changed. And so the repressor protein is has the same kind of allosteric property we talked about last time, only in this case, when you aren't bound by the second molecule, which is going to be down here, the repressor protein is not able to bind DNA. So remember back here, the normal state is this repressor can bind DNA unless the inducer is there, which makes it so it can't. This repressor cannot bind DNA under normal conditions, and so the operon is on. Now, if this molecule will bind to the second site there, that opens up the DNA binding domain. The repressor can come onto the operator, and the same scenario happens as happened with the previous example. Transcription is turned off. So remember back here now, the second molecule, the inducer binding, is going to be the first thing in the biosynthetic pathway. Here in repressible, it's going to be the last thing in the biosynthetic pathway. Yes? This one is going to be the lac operon here. And we'll talk about that. I'll get to that a little bit later today. So if you've talked about the lac operon, if you've taken microbiology, I know that all the instructors there talk about the lac operon. The lac operon is going to be negative inducible, but it also has a separate activator protein that's kind of a twist on that. And we get to that in just a bit. All right, so I always hesitate to ask this because students are less fix-it now than they used to, but do any of you guys know what if you lift the lid on the back of your toilet, do you guys know what that looks like back there? Yes? So when you flush the toilet, the water goes down and then your water supply from your house fills it back up, but then it has to stop, right? So how does it know? How does it know? I mean, it's not intelligent, so how does the water supply get shut off when there's enough? Yeah, there's some kind of either an arm with a float that goes like this, or in some of the more modern ones, there's a little floater on a tower that goes up and down, but the water level in the back of the toilet interacts with the float, and when there's enough, it moves it up and a valve shuts off. You can think of the negative repressible in the same way. So instead of the precursor protein in the pathway being what interacts with the repressor, here you have the product. So when there's enough product, it will bind to the second site on the repressor. That causes an allosteric shape change that in this case allows the repressor to bind to the operator and turn off transcription. It's the same stoichiometry we talked about before. The more product you have, the more repressor is bound and therefore able to bind to the DNA. So if you've got a whole lot of product here, almost all the repressor protein in your cell is going to be bound. It's all going to be on the operator. The RNA polymerase can't get in there, and then transcription can't happen. And same thing that happens with the negative inducible operon, only in reverse here. As the amount of product gets used up by the cell's going to want to make more, right? So as this gets used up, it comes off the second site there. As soon as it comes off the second site, this DNA binding domain closes up and the repressor is no longer able to bind, and there you go. Kind of like, so when there's a lot of product present, it's kind of like when the toilet is full and it shuts off, but as soon as you flush it, you're using up the water in the tank, and so then your mechanism that senses how much water is there calls for the tank to be filled again. These are all four of these are homeostasis mechanisms. So there's this sensing mechanism that controls transcription in all four of these operon situations is responsive to levels of either product or precursor protein in the cell, and will keep those levels at what they're supposed to be. All right, now these second two or final two are described in the book, but the 6th edition of the book, if you have the newest one and not the sort of lavender older one, don't have these pictures, but I think it's important to see what this actually looks like to help you understand. So now we're going to talk about the two examples of positive. This means that instead of a repressor protein, the regulator is going to be an activator. So positive inducible, Because it said inducible, that means that the operon is off and not shown here, but you're going to have all the genes over here that are going to get transcribed. A second gene is going to make a regulator protein that in this case is an activator protein, but it's not going to be able to bind DNA unless a second molecule binds to it. In this case, because you're turning on transcription, the second molecule that binds here is going to be a precursor product. So precursor is present in sufficient quantity, you get binding of the activator, now the DNA can get bound, and in this case, instead of binding the operator, which is downstream from the promoter where the RNA polymerase binds, here the activator protein is going to bind a little bit upstream, so a little bit 5' from the -10 and -35 sequences. And the point here is that the activator protein's job is to help recruit the RNA polymerase. Remember back when we talked about the basic transcriptional mechanisms earlier on, we talked about transcription initiation. We talked about consensus in the -10 and -35 sequences. And we said that sigma factor really likes to have certain sequences there. And the closer you are to those DNA sequences, the stronger that sigma factor is going to be able to bind and bring in RNA polymerase. And in some cases, those -10 and -35 sequences differ a little bit from the consensus. And in those cases, yes, sigma factor can recognize where the -10 and -35 is in here, but the binding is too weak to get a lot of transcription, or any, depending on the promoter. And so in this case, having the activator protein tethered nearby on the DNA will interact with some part of the RNA polymerase and help to make the binding stronger. Once the RNA polymerase is in place, transcription can start just like we talked about earlier on, and then just like the core RNA polymerase, the two alphas, the beta prime, and the omega, leaves sigma and rolls on down the line here. Once transcription is initiated, in addition to leaving sigma at the transcription start site, this transcriptional activator stays there bound to its sequence as well and can serve to bring in another RNA polymerase. All right, and then the final mechanism is a positive repressible operon. And so in this case, what happens is the activator protein, the regulator here, is always able to bind unless you get binding by a second molecule. And because we're turning off the operon when the change happens, it's going to be the end product that does it. And so instead of trying to memorize these, you've heard this before, right? Instead of trying to memorize all 4 of these, learn how they work. You can think your way through this. If I didn't give you this picture, and all I said was you have a positive repressible operon, you should be able to say, okay, it's repressible, that means it's always on. It's positive, that means there's a transcriptional activator involved. And so how are we going to figure this out? If it's always on and there's an activator, then that must mean the activator can bind under normal conditions, and it's only if you bind the second molecule that the activator becomes inactive, because it's going to then not be able to bind and you won't get transcription. So which would be the molecule that would cause transcription to turn off? That's always going to be the end product here, right? Because if you have enough of this end product, then you don't need the genes to be transcribed anymore and transcription can be turned off. It would make no sense for the beginning product to do this. What if that were the case? If you have a lot of this beginning product, you get binding to the activator and it pops off, that makes no sense. That means if you have if you have the precursor protein here, you would turn transcription off, and that makes no sense. So with all of these, if you just learn the four things, repressible, inducible, and positive, and negative, well, negative was the earlier ones, then you can figure it out, right? Yes? Is this going to be an essay question on the exam? Well, I don't give out the questions in advance, but I mean, you know me from earlier times. Whether it's an essay question on the, I'll tell you, whether it's an essay question on the exam or whether it's some of the multiple choice questions, I guarantee there will be, you will need to know all four of these and how they work. So it's going to be on the exam somewhere. As you've seen on the first three tests, there's not much. I try to include everything I've covered in class somewhere on the exam, whether it's in the essay questions or in the multiple choice parts. So yes, you have to know it. All right, so now we're going to talk about a real world situation here, and we're going to bring in a little twist or nuance to the situation. So E. coli was one of the first model genetic organisms. It's easy to grow, easy to make mutants. And so scientists were figuring out how nutritional needs were met in this organism. So in order to get energy, no matter what you start with, it's got to get converted into glucose, because glucose goes into, feeds into glycolysis and the rest of the energy pathway there. Remember, well, I was going to say remember when you were a kid. Remember right now. Or think of a kid you know, right? There's multiple things you can eat to get nutrition. But the kid, what would happen if in front of you or in front of a kid somebody stuck down two dishes and one of them had ice cream in it, which is my glucose example, or the other one had Brussels sprouts in it or broccoli or something else like that? Both would provide nutrition, but given a choice, everybody would want the ice cream, maybe, right? Glucose feeds directly into glycolysis. Organisms have the ability to break down lots of other things and convert them into glucose and then feed them into glycolysis. But if glucose is present and you're fighting for your life again, in the population, in whatever environment you're in, if glucose is present, you want to use that. You only want to expend the extra energy to use other sugars, say, like lactose, if there's no glucose present. So the E. coli lac operon is going to be used for using lactose as a sugar or as an energy source. But two questions have to get asked and answered. So first, the E. coli will only use lactose if no glucose is present. Like I just said, glucose is simple. Why convert lactose into glucose if you don't have to? So if glucose is present, you want to use that directly. And second, lactose must be present. If there's no glucose, you don't want to make the genes to use all the other different kinds of disaccharides and other sugars and every other energy source that E. coli can use to make energy unless it's there. Again, making it wasting energy doing that. All right, so like I said earlier in response to Emily's question, the lac operon is an example of a negative inducible operon. But there's also a separate activator protein that we'll see there. There's two things because two questions have to be answered. All right, before lactose can be used by the cell, it's got to be brought into the cell by a permease. So this is just a channel in the membrane that's able to transport lactose into the cell. And then lactose has to be able to, lactose, which is a disaccharide here, two 6-carbon sugars connected, has to be broken down into its constituent components, glucose and galactose. So glucose can be used directly, and then galactose has to be transformed into glucose. So in order to use lactose, you have to bring it in the cell, bust it up into galactose and glucose, and then make galactose into glucose to use that. So there's a lot of energy expenditure involved. That's why if glucose is present, you're just going to use that. There's 3 enzymes in the lac operon, as we'll see. One of them is going to encode the permease to allow lactose to be brought in the cell, and second one is going to encode beta-galactosidase. So beta-galactosidase is able to metabolize lactose or break lactose down into galactose and glucose. But it's a weird enzyme because it can alternatively break lactose down directly into galactose and glucose, or it can rearrange lactose into a still connected disaccharide called allolactose, and then that can be broken down into galactose and glucose. This distinction is going to be really important because instead of lactose being the inducer that's going to relieve repression, it's actually allolactose. So if beta-galactosidase just did this conversion here directly, the cell would be out of luck. All right, so we're going to talk about answering the is lactose present first, and we're going to tackle the question of is glucose present after this. So remember in this drawing, this looks like the lac repressor protein is very close. It's not, it's somewhere pretty far away in the E. coli genome. You'll notice that all the genes are coded here by names. So LAC-Z, LAC-Y, LAC-A, LAC-I. There's going to be LAC-P, LAC-O. So I'm not going to make you know all this stuff. The reason they have all these funny names for everything that I'm going to explain in just a second is because when the scientists were first learning about how this worked, it was before DNA structure was known, I think, and before actually it was after DNA structure was known, but before sequencing existed. So they did not know whether what they were looking at were genes, were DNA sequences, were proteins, whatever. So they had a mutation that made it so that lactose couldn't be used by the cell. But they didn't know whether the mutation, for example, was in one of these proteins or in the promoter so that the RNA polymerase couldn't bind, or was in the operator sequence so that the repressor couldn't bind. They had no idea. All right. So LAC-Z gene encodes beta-galactosidase, the enzyme we talked about here. The LAC-Y gene encodes the permease, and so you can just know them by those names and not have to memorize the Z, Y, and A. LAC-A encodes an enzyme called a transacetylase that we're still not sure exactly what it does, but we think it helps to get rid of some toxic byproducts of lactose metabolism. But the jury is still out on that one. All right. So because this is an inducible operon, that means that the lac repressor is present all the time, and under normal conditions can bind to the operator, and therefore the RNA polymerase cannot come in here and transcription is off if lactose is absent. So this is regardless of whether glucose is present or not, because even if glucose is absent, Unless lactose is present, you don't want to turn these on. All right, so what happens if lactose is present? Lactose is the first molecule there in the pathway that's going to get converted into glucose and galactose by the beta-galactosidase enzyme. Lactose is also converted into allolactose. That's what binds to, allolactose will bind to this second site on the lac repressor. It will change the shape of the repressor so that the DNA binding domain doesn't work anymore, and it pops off and it allows the RNA polymerase in. And it allows transcription to take place. This isn't as clear-cut as the negative inducible we talked about earlier. Because even though the RNA polymerase is able to get in there now, it's sort of a somewhat weak transcriptional activation. That promoter does not have a consensus sequence that is what it should be. And so it's going to need, the polymerase is going to need some help binding the promoter. But at least this relieves the repression. All right, so now we've talked about answering the question, is lactose present, yes or no? And we move on to the question of glucose. First, I want to show you a little bit about what this looks like over here. So I think I mentioned last time, but if I didn't, the operator sequence, which is over here, that's bound by the lac repressor, actually doesn't cover up the -10 and -35 consensus sequences. It's somewhere else. But even though the -10 and -35 that interact with sigma subunit of the polymerase are not blocked directly, remember we said that the polymerase covers from way up here to way down there, like down to like +20 or something. So the presence of the repressor here in the operator sequence is enough to prevent the polymerase from getting close enough there to activate to get transcription started. This is a picture I got online. So the picture I showed you last time at the beginning of this chapter, it's really cool to see crystal structures of some of these proteins so you can kind of see how it works. So right up here, this yellow corkscrew is DNA, so the helix is going in and out of the board toward you and away from you. And up here are the DNA binding domains. This whole thing, everything that's not yellow, is the lac repressor protein. And so here are the DNA binding domains up here. That's the first site we talked about. And down here is the second site. These little molecules down here are the regulatory domains. And you can see the green there. If you're going to bind allolactose, that will bind in each of these domains, and that will change what this red and this blue domain look like. That will also change what the DNA binding domains up here look like so that they release the DNA and can't bind it anymore. So the presence or absence of the green molecules down here in the regulatory domain changes the shape of the whole protein so that if allolactose is bound down here, the DNA binding domains are not able to interact with the DNA and stick onto the operator sequence there. Too complicated for this class, but there's also a tetramerization domain. So you can see this is a dimer. Here's the red is one piece, and the blue is another piece. So these are each separate polypeptide chains. This will also make a tetramer, so a 4-part, but too much for this. We talk about that in molecular genetics, but I just wanted to show you the structure here because at least for me, it helps a lot to see, instead of little blobs that have little U-shaped openings on them, this is the actual structure that was done using protein crystallography, or actually it's protein and DNA crystallography. And remember I said when I showed this at the beginning, this is similar to what Rosalind Franklin did to figure out the structure of DNA, except that now crystals are hit with a beam of electrons instead of an X-ray source like she used. All right, so now we're going to answer the question about, is glucose present or not? We're going to introduce another protein called CAP. CAP, like the repressor, has two binding sites. One binding site is able to bind to DNA, and the other one binds cyclic AMP. See, everything's coming back from introductory biology. You thought that the second messenger was never going to come back. So when glucose is low in the cell, the reporter cyclic AMP level is high. So the absence of glucose results in a high cyclic AMP activity, and CAP protein is an activator that can bind cyclic AMP. When CAP is bound to cyclic AMP, it can interact with its site on the promoter and help recruit the RNA polymerase, like we talked about for other activator proteins. So, in order for this to be turned on now, the lac repressor is gone. That means lactose is there. Allolactose is bound to the repressor, and so the repressor is off the operator, so the promoter is able for RNA polymerase to come in. And then because glucose is low, cyclic AMP is high, so the CAP protein is able to bind here. So you got two questions being answered. However, when glucose level is high in the cell, the corresponding level of cyclic AMP dips way low. And even though the same amount of CAP is there, now very little of the CAP will be bound to cyclic AMP, and if it's not bound to cyclic AMP, then CAP is not able to bind here to its site. And if it's not bound, it can't help recruit the polymerase. Right? So even though lactose is present and the repressor is gone, RNA polymerase can't bind here more than a little bit. So yes, you'll get a little expression of these, but not too much. It's not that much of a waste. So this phenomenon is called catabolite repression. So catabolic pathways are those in which something is broken down in the cell. If glucose is present, glucose is a catabolite. It's something that's going to be broken down in the cell. If glucose is present, the cell doesn't want to use any other energy source because glucose is the easiest one to use. You get the most bang for your buck. So no matter whether it's present or not, that's it. It's not used. All right. And so this is a sort of a modularity where you have two different questions being answered. So in addition to the lac operon, there are CAP binding sites on many promoters in E. coli that are used on operons to use many different kinds of sugars. So in every one of these cases, there's going to be some other mechanism. So this is lactose. What happens if it's another disaccharide? There's going to be a regulation of the genes required to use that one. Is that other sugar there? Yes or no? Like if maltose is there. Is maltose there? If yes, then you want the operon to be turned on. But just like for lactose, in order to use maltose, in addition to maltose being there, you want there to be no glucose. So I can't remember the exact, I think it's over 100 promoters in E. coli that the CAP binding site is on, and this is an evolutionary mechanism. So once you've evolved the ability to sense, is glucose there, yes or no, then that gets taken and used many other places because the cell could just copy and paste that DNA through duplications, and it doesn't have to reinvent the wheel, so to speak. All right, so that is all I'm interested in having you know from chapter 16. Once the class is over, there's lots of interesting stuff in there that we don't have time to do. So in addition to regulation of transcription in this way, there's also a lot of interesting information about regulation of transcription by what the 5' end of the messenger RNAs look like. That's something that, if it interests you, you should go on and read. But I do not have time in this class to go through it. All right. Any questions before I go on to chapter 17? All right. And just like chapter 16, chapter 17, which is eukaryotic transcriptional regulation, is fairly just examples that will a little bit how this works without having to have you memorize all the pathways that are in that chapter. Remember way back in chapter 11 we talked about DNA packaging and histones? Well, they're going to come back. So originally, as I said back then, histones were originally thought to be just involved in DNA packaging. But the big revolution in the late 1990s and the 2000s is that histones are intimately involved in transcriptional regulation by allowing or denying access of transcription factors to the DNA. And this is the nucleosome here. The black is DNA that's wrapped around it. And remember we said that the histone is an octamer. So there's going to be 2 histone H2As, 2 H2Bs, and then a tetramer that contains H3 and H4 like that. And this geeky scientist's birthday cake is actually a really cool example that shows better than the pictures in the book does those histone N-terminal tails that I talked about. So here's the part of the histone that is involved in binding DNA generally, and then here are all the little N-terminal or amino-terminal tails coming out there. And as we'll see, these are going to be involved with having a bunch of little modifications put on there, as you can see here. And we'll talk about that in just a second. All right, so eukaryotes, in addition to are transcription factors present or not, which is the primary mechanism of prokaryotic regulation, are also considering changes in chromatin structure. And we also talked previously about epigenetics. So remember, we talked about epigenetics in chapter 5, and that is a heritable change in phenotype that does not result in a change to like the DNA bases. So when we talk about this in a little bit, we're going to say like changes to what modifications are on the histone tails are going to be important. And in previous chapters, we already discussed about one epigenetics modification. Do you remember when we talked about methylation of DNA? In the DNA structure chapter, chapter 10, and if you put that CH3 group on the C of a CG dinucleotide, that signals to the cell that that gene is to be transcriptionally turned off. We're going to get more of that here. So eukaryotes, like prokaryotes, do regulate through transcription initiation. We're going to talk about RNA processing and degradation. So we already talked about RNA splicing, where you take the introns out of a pre-messenger RNA, and we talked about alternative splicing. That's what we're talking about here, where you pick and choose certain exons to include in the final messenger RNA. We're also going to talk about a Nobel Prize-winning phenomenon that was discovered in 1998 called RNA interference. that has kind of revolutionized our understanding of biology. I talked a little bit about this before, and it's in black instead of blue there because we don't have time to go here, but if you take cell biology, and I highly recommend it, Dr. O'Neill is great in that class, there is regulation at the level of the protein. So both whether a protein is turned on or off based on modifications like phosphate groups being added or removed, and also whether proteins are taken to the 26S proteasome and shredded or degraded. So this is level of regulation all the way down after the protein is already made, but we don't have time to do it here. Okay, so I mentioned this a little bit when we talked about in chapter 11 about DNA packaging. In addition to compacting the DNA, putting DNA into nucleosomes can obscure transcription factor binding sites. So remember we said there was some of the DNA that's wrapped tightly around the nucleosome, and then there was this linker DNA that goes between them. We also talked about the fact that binding of DNA to the nucleosome is not sequence specific. So the histone proteins have positive charge on and interact with the negative charge of the sugar phosphate backbone. That allows nucleosomes to be positioned or moved around. This is accomplished by chromatin remodeling complexes. Also, a hot topic of scientific investigation in the last 20 years or so. So chromatin remodeling complexes are capable of either removing a histone or moving histones farther apart, lots of different mechanisms for this, and exposing transcription factor binding sites at a certain gene. So up here, this is going to be what we talked about before as being closed chromatin, where even if so here's a transcription factor binding site. Even if that transcription factor is present, if the DNA is closed up like that, it can't bind, and so transcription can't be turned on. This is another way that we talked about before, how even though we have not that many more genes than the C. elegans nematodes do, we can do a lot more with what we have because we don't have to make separate transcriptional regulatory proteins for all the different conditions. We can shut a gene off just by locking it up in chromatin and making the transcription factor binding sites inaccessible. Once the site is accessible, transcription factor can bind to it, help recruit the RNA polymerase, and then transcription gets started. So they changed this picture between the 5th edition and the 6th edition of the book, and you'll see in a couple slides from now the current one. I like this picture a lot better because it shows here is the octamer, here is the DNA wrapped around it, and under normal conditions those histone tails that were shown sticking out from the cake are positively charged, and so they will grip the DNA more tightly and sort of lock the histone down into place. But a global mechanism of change called acetylation is able to change that. So these tails are positively charged, grab the DNA tightly, closed chromatin, no transcription, and the nucleosomes can't move very well at all. But if you get acetylation added, you get acetyl groups added to the side chains on the N-terminal tails. That relieves or gets rid of the positive charge. They don't bind the DNA as well. And yes, the histone still maintains its grip on DNA, but it was what we talked about when we talked about histones and their ability to move. Remember, like when you turn your steering wheel on the car, and then when you're coming out of the corner, instead of going back around like this, you just sort of like let it go, right? And the steering wheel moves in your hands, but you still got a hold of it. That's the same thing that's happening here. All right, so I added this slide. I added this slide and reposted it on Blackboard. You don't have to know this at all, but I just thought that this would be a cool thing to see as far as this is what it is for those who want to see the actual chemistry. So here's lysine. It's a positively charged amino acid because it's got an amino group on the end. But if you add an acetyl group, so there's the C double bond O and the CH3 on the end of that, now this nitrogen does not have a positive charge on it anymore. So a histone acetyl transferase enzyme will make this happen, and the HDAC or histone deacetylation will remove it and go back to that. This is the picture from the book that they changed to. You can see maybe why I don't like it. Because here and down here, the tails are both, in both cases, are like sticking away from the DNA, right? This is not gripping the DNA tightly and this is loosening it. I mean, this is a good example of just pretend these were locked down around the DNA. It looks like somebody just made those little squigglies and cut and paste them all in there because they were in a hurry. So when you see this in the book, think of this instead. Think of how these are gripping the DNA, and that's what should be happening up here. So acetylation is a global mechanism. So if the histones in a certain area are generally acetylated, that will make that whole area more open chromatin. But like a lot of things we talk about in here, it's more complicated, right? So it turns out that in addition to global or just generally acetylating all the lysine amino acids to get rid of the positive charges, there are other marks that can happen. You can get acetylation of specific amino acids in the N-terminal tails, also methylation and phosphorylation. So this became known as the histone code, and I'm going to show, this is my prediction. Every year I put Dave Alice's picture up on there, and I wonder why he has not got the Nobel Prize yet, because it would be a fairly big crime if he did not get the Nobel Prize for figuring all this stuff out. So this is what I mean by the histone code, and here's a bigger picture of it. This is like the chalkboard from their lab. No, you don't need to know any of this. Like here's histone H3, this is the amino terminal. Look, here's all these single letters here are the letters for all the amino acids. All the stuff that's written above and below there are specific changes that can happen to specific amino acids. And each set of changes, when they're put together, form this histone code that will tell what's supposed to happen. And so here's histone H3, H4, H2B, and here's a couple of other. I think this is the end of H3 from up there, and this is the end of H4 from there. But this was a fairly large amount of work. So for example, I mean, you don't have to know this, but like here, lysine 4 right here, if you put a methyl group on there, that will lead to a protein binding to it, and something happens in the cell. The way that Dave Alice, and I'm going to put his picture up here again, figured this out was amazing. Does everybody know, have you guys taken immunology, or some of you, it's like, okay, well, you guys have all had a flu shot, right? You need to get a flu shot. You could go over to the health center and get your flu shot. You've all had a measles shot, right? Please say yes. Okay, when you get a vaccination against something, what happens? What is that? What's a vaccination do? Right, you make antibodies, right? So if you get an immunization, they'll inject you with either a killed or a weakened version of either the bacteria or the virus that you're trying to get protection against, and your immune system will recognize those proteins and make antibodies against them, so that if you ever see that again, they'll attack it. Making antibodies is a big tool for molecular biologists. So each one of these things, they've got an antibody to. For example, they can take histone H3 and methylate lysine 4, and they took that and injected it into rabbits. And then after a while you get a booster shot to the rabbit, just like you do, and pretty soon you'll get antibodies. And they found, either rabbits or mice, I don't know which they did for this one, but they were able to get an antibody that can recognize a methylated lysine K4. The difference between a regular lysine and this one is a methyl group. The antibody recognizes just an extra CH3 on the end of that. It's pretty amazing. And so then they can use these antibodies in cells and show in chromosomes whether that methylation event has happened or not, and see what happens in there. Yeah. Someone stole the purple and the brown marker. Well, they could just be out of the picture. Anyway, but they did that for all of these things. Of course, Dave Alice is at Rockefeller University, which does anybody know what that is? It's in New York City. It doesn't have undergraduates. It's all graduate students and postdocs, and it's for high flyers, let's say. And so they got scads and scads of money. All right, but this is pretty cool, like that their lab went through and figured out all this stuff. Nobody knew that this was a mechanism, but he's like, I wonder if that does anything. And then they did all this investigation. Of course, once he started it, everybody jumped on the bandwagon and kept going with it. But that's the thinking that, well, should get you a Nobel Prize anyway. All right. All right, so here's an example that the book gives using the little mustard plant, Arabidopsis, that we talked about as sort of like the fruit fly of the plant world. When I talked about model genetic organisms way back in chapter 3, I mentioned this one as it's very small, so the rosette of leaves is only about that big, and it puts up a little flower stalk. It's about that yay high. And the most important thing about Arabidopsis is it has a very quick generation time, only like I think 2 months from the seed germinating to having other seeds, and it grows really nicely in environmental chambers under fluorescent lights. So just like our growth chamber up there that we grow the C. elegans worms in, that could be an Arabidopsis growth chamber. All we'd have to do is add some fluorescent lights to it on a timer. So if you have the old 5th edition book, this is a mistake. It says transcriptional activator protein. This is not. It's a repressor protein. If you have the 6th edition book, this is corrected and it says regulator protein there. But stupidly, when I tried to sub this out, I went and figure 17.3 that comes in the instructor materials for the 6th edition still has the old 5th edition thing in there. So I put that in there. If you have the 5th edition, cross this out and write repressor in your book. Otherwise, what I'm about to say will make no sense at all. So flowering time in flowering plants is very important. And it's one of the issues that is of greatest importance to plant and insect survival with climate change happening. So as global warming occurs, spring is happening earlier and earlier. And so things that used to happen in June may happen in the end of the end of April or at some time in May. Plants and insects have co-evolved to have the flowers on the plants be blooming at the same time that the insects that need to pollinate them are around. And so sometimes you can have problems in that if climate change makes the plants start blooming earlier and the flowers come out, but the insects don't also evolve to hatch from the ground or come out from their larval state earlier, then the plant can come out, the flower's there, but the pollinator is not. And so that's a problem for the plant because the insect that it needs to pollinate, it's not there. It's also a problem for the insect if it comes out of the ground 2 weeks later and the flowers on the plant are gone. You know, in some cases, insects are evolved to use specific flowers on specific plants. This is an example of regulation by chromatin structure. It's also an example of a regulatory cascade that you'll see more of if you go into cell biology. It's a fairly simple one because that's why they picked it for the example. But in some cases, regulatory signaling cascades can have 9 or 10 steps or even more. But it's going to be something like this. All right. So, and you do need to know FLC and FLD because there's not good names for these proteins other than them. So under normal conditions, the area of chromatin where the FLC gene is open. So that allows transcription to take place, the mRNA is produced, translation takes place, and that makes a protein that is a repressor that negatively regulates flowering in these plants. So there's not a signal that says flower now. There's a signal that says don't flower now, right? If you mutated this gene, the flowering regulation in Arabidopsis would be way off. There'd be no repressor, and they would flower much earlier than they're supposed to. So how do you actually get flowering? In this case, at some time when flowering is supposed to happen, the FLD gene is changed. FLD is transcribed into messenger RNA, translated into protein, and this is a deacetylase enzyme. So remember we talked about before, acetylated versus deacetylated. So acetyl groups do what to the charges of the histone tails? Take them away. So instead of being positively charged, they're neutral. If histone tails are positively charged, what's that mean as far as their gripping of the DNA? It's pretty strong. And so if you take that away, that makes it weaker. So this DNA is acetylated normally. It's open. The deacetylase enzyme comes in and specific to this region removes the acetyl groups, or removes the acetyl groups from around here. That leads to the positive charge coming back, and you go from an open to a closed chromatin, right? The N-terminal tails are gripping the DNA more strongly, and chromatin becomes inaccessible. Because the chromatin becomes inaccessible, then the FLC gene can no longer be transcribed and translated into protein. You get no repressor, and therefore flowering occurs. It's one of those, what we talked about last time, the enemy of my enemy is my friend sort of situations, right? So the deacetylase enzyme represses the repressor. Doesn't do it directly, but it eliminates the expression of the repressor because it shuts down the chromatin from open to closed conformation. You don't make any more repressor protein, and now there's nothing stopping flowering from occurring. I do not know. So there's going to be, it's probably, so there's going to be some temporal specific transcription regulatory event that leads to this deacetylase being, transcription of the deacetylase gets turned on at the right time. That regulatory mechanism, I don't know. But it's going to be something like that. And this is just a two-step thing. A lot of these cascades are, like I said, have not 8, 9, 10 different levels. one thing represses another one, which is an activator of this, which represses the third thing, and it's a whole bunch of pluses and minuses. So that's regulation at the level of accessibility of the chromatin. This is going to be the same thing. So methylation of CpG dinucleotides does the same thing. We showed this earlier back in the DNA chapter, chapter 10. But a CpG dinucleotide is just a C that's next to a G. The P is just the phosphate between them. And we talked then about why it's important for this regulation to be only on CpGs, not just Cs that don't have a G next to them. And that's because the anti-parallel way that DNA is set up, that wherever there's a C, there's going to be a G across from it. And wherever there's a G, there's going to be a C across from it. That means that both strands of the DNA will have the methylation on them. If you just had C and then these weren't Gs, then there might not be Cs on the opposite strand. But having a CG, CG, CG arrangement means that there's going to be Cs and Gs over there. So methylation of Cs in CpG dinucleotides has been known for a long time to signal transcriptional silence. Proteins recognize this and then do the histone rearrangements to make the chromatin inaccessible. And I could not resist throwing in this paper from a famous scientist who was working back in the Pleistocene era. Back when I was in graduate school, I was working on transcriptional regulation of genes that are only expressed during mouse spermatogenesis. So I was still doing reproductive biology, but I was doing it in a different species. And we were looking at transcription factor binding sites. So these are sites that are recognized by specific transcription factors. You don't have to worry about what they are or anything like that. And we wanted to look at the methylation status. So we did a special biochemistry technique that I had to do in the fume hood because it'll kill you if it gets out. So it was a fun experiment to do. That we took the DNA isolated from testes and liver, which is any tissue that's not testes, and did this biochemical conversion reaction in which unmethylated Cs are converted into Ts. But if the C has a methyl group on it, the chemical reaction can't work, and it stays a C. All right, so here are a bunch of T's. Every one of these T's that is circled used to be a C. So the reaction converted all these guys from C's to T's. The reaction did not convert these C's to T's in the liver. So if you look, this one's kind of out in the middle of nowhere, but each of these transcription factor binding sites has a C in it that is methylated in the liver but not in the testis. This gene is expressed in the testis but not in the liver, and by figuring out that these C's, and here's one in the middle, are methylated, that means that the methylation could be partially responsible for this, because these transcription factors that recognize these sites might be present, and a lot of them are, in the liver as well, but here the DNA is methylated, so the chromatin is compacted and they can't access it. I was unable to find, because this is ancient, I lost my sequence traces, so these should be in color. The reason why this is N, you can see two little peaks here, so that's like 50% C and 50% T, so half of it was converted, so it's it's a little bit resistance. But this demonstrates that, you know, methylation in a non-expressing tissue is part of the reason for testis-specific expression there. So I talked about epigenetics, and I said epigenetics are these markings that are present that do not require a DNA base change, but they are also heritable. That means they can be passed down from one generation to the next. And at least for methylation, this is how that works. So, and it's also important that you have methylation on both strands like I talked about earlier. So remember back from the DNA replication chapter, you know, here's a replication fork. It's opening up, it's moving toward the left, and you have the original DNA strands here and then the new DNA being synthesized. The new DNA being synthesized is not going to have the methylation on any of the C's that this area has been marked with. The reason for that is because the polymerase doesn't know to do that. It just goes through and messes around, and what you end up with is this situation here. This is referred to as being hemimethylated. So one strand is methylated and the other strand is not. And then what happens right after transcription is a methylase enzyme or methyltransferase enzyme will come in, and it's able to read methylation on the old strand, and it will put it on the new strand. This happens every time mitosis occurs in the cell, except when there's a specific signal to change it. Remember our other epigenetic example was for the immunoglobulin-like growth factor gene that is marked so that in males that gene is active, but in females it's not, because males want their offspring to be bigger and females want their uterus not to explode so they can make more babies in the future. That marking is reset in the germline. So in the males, even though one of your chromosomes is marked to be on and one's marked to be off, they're reset so they're both on, and in females it was the opposite, right? They're both marked to be off. Well, this can be reset to be methylated or not if, you know, if that's the case. All right. We only have a couple more minutes left, so I'm going to start this, and then I want you to finish reading this, finish reading about eukaryotic transcriptional regulation. We'll go over this on Tuesday when we get back. So the plan is we're just going to finish out this chapter, which I don't have that many slides left here to do because I've picked and chosen, and then we'll use the rest of Tuesday as a review session. And I can have another review session like we've done for the earlier exams if you want to later on, you know, when the exam gets closer, like maybe Monday the 2nd or something. If you just tell me, you know, what time and where, like we've done before, we can do that. But on Tuesday after Thanksgiving, come, you know, read all this stuff over and come in with a bunch of questions, and we can have a review session there and ask the questions. All right. So eukaryotic transcription regulation, similar and different than prokaryotic. Remember, the RNA polymerase, instead of being brought to the transcription start site by a single protein, the sigma factor, in eukaryotes you have all these different transcription factors that work in concert to do the same thing. So as we talked about for the last exam, transcription factor 2D has the TATA binding protein in it. Everything comes together. And we also talked previously, and this is not to scale at all, about the mediator complex. Remember, it's not to scale because there's about 30 different proteins, and that will interact between the basal transcription machinery here and either enhancers or silencers that are located far away, and in some cases very far away, like 75,000 to 100,000 bases away from where the transcription start site is here. Enhancers are going to bind transcriptional activator proteins, not shown here, but silencer sequences will bind transcriptional repressor proteins. These will have their effect by interacting with mediator, which then will influence the general transcription factors there. All right, so it is now 10:40. I will see you guys if you have not done the lab yet. I'll see you guys there. Can you hold on just a second? If you are Tuesday lab people, there was, and I think I put this on the announcement, but there were two groups that thought they were gel number 2. The second group, where I took two pictures, one brighter than the other, the 2A and 2B, if that was you, you really are gel 2. If you thought you were gel 2, but there were not two pictures taken of your gel, then you are now gel 3. Gel 1 was just gel 1, and those are all on the website for you to get. All right, see you later. Yeah. And, oh, did you tell me at the time?